So we were just talking about the neuromuscular junction, and now let's talk about how the communication from nerve action potential turns into muscle action potential. So, at our neuromuscular junction, the synapse between the motor neuron and the muscle cell, you'll see all this specialization and invagination for these junctional folds that go, and they're a way of increasing the surface area to make sure that the acetylcholine that is released from the nerve terminal provides a signal that's going to depolarize the muscle cell. There will be many active zones, areas where the acetylcholine released. You have this large surface area, and as the acetylcholine is released, then it binds to the acetylcholine receptor, the nicotinic acetylcholine receptor, opening an ion channel and causing a local depolarization. This is called an end-plate potential, and it is not yet an action potential. Most of the ion flow that comes across the acetylcholine receptor will be sodium coming into the cell, but there is some potassium leakage out of the cell at the same time. Enough sodium over potassium comes in so that you get a local depolarization, that end-plate potential. The end-plate potential will then move passively. It's not an action potential yet. It's passive spread, and it will encounter an area of the membrane that is rich in voltage-gated sodium channels. Now, from Dr. K's lectures, you know that if there is a depolarization large enough near a voltage-gated sodium channel, it will open, and that will trigger an action potential. The action potential will then move along the muscle cell membrane, and unfortunately, this diagram in the textbook makes it seem like it's all very close together. It doesn't have to be that close together. The sodium channels will be relatively close to the motor end plate, but then once you leave that area of the motor end plate, the sodium channels are distributed all along the plasma membrane, the sarcolemma, and through the T-tubular network so that that action potential can continue to move along the muscle cell. Action potential then goes down through the transverse tubular network, and in the transverse tubular network, it will encounter a voltage-gated channel called the dihydropyridine receptor. The dihydropyridine receptor is essentially a voltage-gated calcium channel, but that's not what it's doing here. That's very important. It's not what it's doing here. Any voltage-gated channel has a part of its structure that is highly charged and will respond to electrical signal. And so the wave of depolarization that passes this dihydropyridine receptor will cause it to physically move, and because the dihydropyridine receptor has a physical connection with the ryanodine receptor, which is the calcium release channel from the sarcoplasmic reticulum, you will trigger calcium release. So net entry of sodium here through the acetylcholine receptors will trigger muscle action potentials due to voltage-gated sodium channels in the plasma membrane. This network, because of all these perforations, means that action potential then travels through the center of the cell and around every myofibril, triggering calcium release from the sarcoplasmic reticulum around every myofibril. Action potential at the center, how does that affect the muscle cell? So we get a nerve action potential, and if we have an electrode here, we can measure the nerve action potential, show that depolarization, show it's very sharp and short. This will trigger a muscle fiber action potential, which is a bit broader, which will lead to a contraction. So, electrical event in the nerve leads to chemical release of the neuromuscular junction. Chemical release of the neuromuscular junction leads to a end-plate potential at the motor end plate, which then leads to a muscle cell action potential, which travels in both directions along the muscle cell. And I should say now that in most cases you'd find that the motor end plate, or synapse between the muscle and the nerve, is about in the middle of a long fiber, a long muscle fiber, so that it can move towards the end of both ends of the muscle cell and reach the ends at about the same time, so the entire cell activates simultaneously, or essentially simultaneously. Note though, here's the nerve action potential, very short. Slightly after it, you get the muscle cell action potential, slightly longer. Nothing's happening here. This is so-called latent period. This tells you that something has to happen within the muscle cell before you see a response. What's happening, and I've already told you what's happening within the muscle cell, is first the electrical signal very quickly moves throughout the muscle cell, along the plasmalemma, through the transverse tubular network, and then that triggers calcium release. And so during this latent period, you are having a propagation of the electrical signal, and you're getting a release of calcium inside the muscle cell that's going to lead to this contraction phase. Now, this is followed by a relaxation phase, and the next thing that happens is that the calcium, after it's been released, has to be reaccumulated or resequestered, taken out of the cytoplasm, because we've only had a single action potential, which leads to this event, which is called a muscle twitch, and it only lasts between 10 to 100 milliseconds. Very rapid contraction, very rapid relaxation, and this should tell you that a single action potential can only cause a twitch, and since most of our movements require sustained movement over a longer period of time, they're not due to single action potentials, they're due to something else. Alright, excitation contraction coupling. You get an end plate potential. It spreads passively until it encounters nearby conjugated sodium channels. Opening of those sodium channels will trigger, and the potassium channels, of course, would repolarize the cell, will trigger the muscle cell's action potential. If you get a single action potential, you get a twitch, which is a single contraction relaxation cycle. There's a latent period, which is a short delay between the muscle signal and the development of tension, and this is because time is required for calcium release and calcium binding to troponin. We'll talk about troponin in just a moment. Here's troponin on the thin filament. It's a calcium binding protein. It changes its shape when it binds calcium, and that can trigger muscle contraction. Let's talk about that. So we have our action potential moving down the transverse tubular network, going to both ends of the muscle cell, and the movement of the dihydropyridine receptor in the transverse tubular network triggers the opening of the ryanodine receptor in the sarcoplasmic reticulum, allowing calcium, which is in high concentration inside, to flow down its concentration gradient into the sarcoplasm. Calcium is released. As it's released, you start to build calcium concentration within the cytoplasm or sarcoplasm. Now, on the thin filament, you have these troponin complexes. It's actually 3 subunits: T and C, the calcium binding subunit, along with TNT and TNI, which help it to move tropomyosin, which is on the thin filament here. Note the position that tropomyosin is shown here. It's sitting on top of these actin monomers, and that the actin monomers have been drawn in such a way as to have 2 different colors, one a little golden, one probably a little redder. Troponin and tropomyosin are sitting right on top of that gold patch on the actin. This is where myosin needs to bind to actin in order to make force. So, from this position, the troponin-tropomyosin complex is in the way. However, when calcium binds to troponin, it causes tropomyosin to move more into the groove of this actin than filament, this double-stranded alpha helical filament moves off of the outside edge into the groove, opening up that myosin binding site. So calcium is released, it binds to troponin, causes tropomyosin to move on the thin filament. When that happens, tropomyosin and troponin will move out of the way. Myosin can interact with actin, and it can go through what is called a power stroke. It changes its shape. Here it's more perpendicular to the filament axis. Is the filament. It's perpendicular to the filament, and it binds in this orientation and then rotates. And as it rotates, it pulls on the actin thin filament. The actin thin filament moves relative to the myosin thick filament. Now you can see here it's showing you some ADP triphosphate and how they are bound to the myosin head at the beginning of the power stroke and that they will begin to be released as the power stroke begins. We'll talk about that in just a second. But calcium binding to troponin is what causes this movement of tropomyosin from a blocking position to an unblocked position. So here is our contraction cycle. Myosin binds ATP, adenosine triphosphate. This is a high energy molecule. Cleaving the third phosphate from adenosine triphosphate will cause you to create adenosine diphosphate, ADP, and free phosphate. Most enzymes, when they work, they'll do their chemistry and then they release their product so that they can bind another molecule of substrate or the original starting material. Myosin, when it binds ATP, it rapidly hydrolyzes it, and in doing so it changes its shape. Note here when myosin bound ATP, it was kind of in a bent conformation, and then when it hydrolyzed ATP, it moved to a more perpendicular conformation. So sometimes this is referred to as a 45-degree state of myosin, and this is the 90-degree state of myosin. By changing its orientation, myosin stored the energy that came out of that ATP hydrolysis event. It can associate with actin, and unfortunately this diagram does not show the tropomyosin-troponin complex on the thin filament, but only weakly in this conformation because the troponin-tropomyosin is in the way. Now, when calcium enters the cytoplasm and binds to troponin and tropomyosin and moves them out of the way, then myosin can bind more strongly to actin, and it will first release phosphate as it begins this power stroke or swiveling of the head, and then it will continue that power stroke and it will also release ADP. At the end of the power stroke, the myosin is very tightly bound to the actin thin filament, and it will stay that way in what's called a Riker state. Now, you may have heard of the state of rigor mortis. That's the same thing. Riker means that the myosin bound to the actin is locked there in a very tight conformation, very stiff. It requires another ATP to release the myosin from the thin filament, and then as long as the calcium concentration is high, you can go through this cycle again and again and again. And as each time you go through it, the myosin will pull a little bit on the thin filament and the tension will build up. So, let's go back to our overview. We have calcium release is bound to the thin filament. Myosin is binding after the tropomyosin moves, and then it goes through its power stroke, executes a power stroke. The thin filaments will move relative to the thick filaments towards the center of the sarcomere. Now, the calcium that has been released from the sarcoplasmic reticulum has to be reaccumulated. And there is a pump protein called the calcium ATPase, sarcoplasmic calcium ATPase, that is in the membrane of the sarcoplasmic reticulum. And as soon as calcium is released, it starts using the energy from ATP hydrolysis to pump calcium against its concentration gradient from the cytoplasm back into the sarcoplasmic reticulum. This pumping action will reduce the concentration of calcium in the cytoplasm. Reducing the concentration of calcium in the cytoplasm will then result in relaxation because the calcium will be released from the tropomyosin and troponin complex. That will allow troponin to go back to that position that blocks myosin. The myosin will have switched to its weak binding form because it bound another molecule of ATP. So then it won't be able to lock on and hold on to the actin in that tight conformation. So calcium is pumped away, you get back into the relaxed state of the muscle cell. This is an overview, and I would expect that you would be able to walk yourself through these steps, either by looking at these diagrams, or if given one of the steps, you know what happens next. So calcium is released, the cross bridge is released, the muscle relaxes. Tropomyosin recovers that binding site, and